My name is Claire Kelly, and I am a senior in the Immunology and Medical Microbiology program at WVU. My research focuses on the characterization of interleukin-6 regulation in neuroinflammation. Interleukin-6, or just IL-6, is a cytokine. Cytokines are used by the immune system to communicate with other components of the immune system. They are proteins created by one immune cell to signal to what's going on to other immune cells. This cytokine is usually only expressed in your cells during conditions of inflammation or other types of stress. When your cells are just in their normal resting state, IL-6 is not being produced. And what we don't understand is how the cell regulates this expression. It needs to be turned off and on according to what's happening inside of your body. So just a little bit of background first. This is just a very simple schematic of protein synthesis. We all know that the DNA holds your information, important messages for your cells. DNA is transcribed into RNA, which encodes the same message as the original DNA, just a different medium now. RNA is then translated into a protein. And this is how IL-6 is made too, from the genetic code into a functional protein. So if you can imagine this line here as being this IL-6 RNA, what we think is happening is that the machinery used to transcribe the mRNA into a protein is using different open reading frames. So for example, if the machinery were going to transcribe just open reading frame A here, it would start at the beginning and it would stop at the end of this blue bar. If you notice below this, this only creates the non-coding region of the protein that we need. We don't fully understand the point of non-coding regions, but we do know that it does not contain the functional IL-6 that we need. So we think that during stressful and inflammatory conditions, the cell is translating IL-6 at this second open reading frame that will give us the IL-6 protein that we need in order for this immune response to work. So what we did was we used a human glioma cell line, and we made different mutants of these cells using the CRISPR-Cas9 gene editing system. What this does is this uses the enzyme Cas9 to cut a targeted area of the genome, which is found by this guide RNA. So cutting this part of the gene, and then the cell will religate it to fix this break in the DNA, creates a mutation. So what we've done is we've generated a handful of mutated cells to see how they produce IL-6 differently. All of these mutations that we have are in the open reading frame of IL-6. So what we did was we have 4 treatment groups. We have the wild type cells, which are the cells that were not mutated. It's just how they naturally are. And we have our mutant. This says mutant 11. We have several different ones that can plug into this methodology. To induce a cellular stress response, we treat with a chemical called thapsigargan, which is used to induce stress, particularly in the cell's endoplasmic reticulum. The endoplasmic reticulum is the part of the cell that is responsible for producing proteins and processing them. to create the functional proteins that our cells need. So what we do after we have our treatment groups is after 4 hours, we will collect the samples. What we do is we actually first isolate the RNA of these cells, and we can test the level of RNA transcripts by then turning it back into DNA and performing RT-qPCR, which is a real-time analysis that allows us to quantify RNA transcript levels. So we do that, and then sometimes we let this treatment go for 24 hours, and then we will perform what is called an enzyme-linked immunosorbent assay, or annotated here as in ELISA. And this allows us to do essentially the same type of thing with protein. So this will allow us to quantify the amount of protein that is being produced by these cells. In our case, obviously, we are trying to quantify specifically the amount of IL-6 protein. So we did this for a few of our mutants that we have. So in panel A, we have the fold change of the mRNA levels of IL-6 produced in one of our mutants. Fold change is just comparing the level of one of these groups compared to a non-treated wild type level. So in this last bar here where it says this is our mutant that's been treated with the thapsigargan for the endoplasmic reticulum stress, this is how many more times than the wild type IL-6 transcripts this is making. So if we look back here, it's making this part of the IL-6, not the full protein, but just the intermediate player. In panel B, we see a similar pattern here for the amount of protein that's being produced. We see a huge increase when we get to our mutant that's been treated for stress. So that tells us that this particular mutant that we've created here produces a lot more IL-6 than just a normal cell when it has been introduced to these stressful conditions. So this data also shows us something neat, that the IL-6 control of the expression is being regulated at this transcriptional level, because we see the same patterns, we get this same type of RNA and protein expression, which means that somewhere before in this transcription process, IL-6 is being regulated. So if we move on to panel A up here, we have again the mRNA fold change, and we see a similar pattern that we saw before. This is a different mutant. These are mutants 11 and 10. They have different mutations in their DNA. Other than that, 11 and 10 are just arbitrary numbers that we use to keep track. So this mutant, again, much more IL-6 transcript or mRNA is being produced under the cell stress conditions. However, when we performed an ELISA to see the amount of protein being produced, we got drastically different results. This is not the case. We see that not a lot of IL-6 is being produced in these stressful conditions of our mutant, but it is in the stressful conditions of just our normal wild type cells. So this possibly shows us that this particular mutant is controlling IL-6 production at the translational step. So there's a lot of mRNA being produced, and somewhere along the way, it's not all getting translated into protein. So what our data shows us so far is that we have different open reading frame mutations that can affect the expression of IL-6 in different ways. We also know We also know that the cellular stress response increases the amount of IL-6 in both our wild type and our mutated cells here, our RNA, but not necessarily with our protein. We also, as I went over, know that these different mutations can affect the outcome at both the transcriptional and the translational level, depending on the specific mutation and what that's doing at a molecular level. So our next step in what I'm working on now is to sequence the IL-6 gene of our different mutants to see exactly what's going on within the genetic code of this protein. So panel A here is just kind of a depiction of what we have. So maybe this is just trying to show you that there are mutations at different places in each mutant. So hypothetically, this one on the top could be mutant 11 and this one on the bottom could be mutant 10. So I'm isolating the DNA and we send them to a core facility for sequencing, and they that will show us exactly the molecular change that has occurred in the DNA that causes these different mutations, which will cause these different levels of expression. I'm also currently working on generating more of this, the RNA and the protein data for more of our mutants. We have much more than 2, and my progress has been a little bit slowed by COVID and other issues, but I'm working on it. And so hopefully this will help us understand how the IL-6 in the inflammatory response is being regulated. This is important because this is a mechanism of disease and inflammation that occurs a lot in neurodegenerative diseases such as multiple sclerosis, ALS, Alzheimer's, or stroke. So this is just all part of our lab's research to better understand the molecular level of neuroinflammation and kind of maybe how we can control it and kind of create a better outcome and maybe even treatment for some of these diseases. So I would like to thank Dr. Gordon Mears and all of my lab mates for helping me with this. And my research is supported and funded by the Immunology and Medical Microbiology Internship through WVU.